201st birthday of Ishwar Chandra Vidyasagar, the great luminary, the great reformer of the India. So, in fact, I said that is Institute of Science, Education and Culture have organized one national seminar to commemorate the 200th birth anniversary of Ishwar Chandra Vidyasagar on February 8, 2020. And this one was national seminar. The topic was Ishwar Chandra Vidyasagar's thoughts on education. The topic was first, secular humanist, educationist and ideas of Ishwar Chandra Vidyasagar, and it was delivered by Professor Priyambada Sarkar of Calcutta University. The second topic was Ishwar Chandra Vidyasagar's role in Bengal Renaissance, and it was delivered by Dr. Pulobi Mukherjee, formerly with Bexhill Memorial Girls College. The third topic was delivered by Professor Chitrabrata Pali, formerly with Bexhill University, and this topic was stages of education in Vidyasagar's thought. The last topic was on the role of Vidyasagar in developing child education and Borno Porichaya, and it was delivered by Dr. Lili Vishwas of the University of Kolkata. So we pay our deep homage to Ishwar Chandra Vidyasagar on his 200th birthday today, and on this auspicious occasion we are going to conduct the 40th annual general meeting of the Institute of Science, Education and Culture. With this brief introduction, I now request Dr. Vandana Burman to please run and present the inaugural address by our President, Professor Dr. Anil Kumar Ghosh. Dr. Burman, please. Dr. Burman is trying to present the audio presentation of Professor Ghosh. It takes time. This is quite normal in this networking issues and all these things. Dr. Burman, is there any problem? Please again trying to present the lecture of Professor Anil Kumar Ghosh. We are having 2 minutes. Meanwhile, I can just tell we are having here 2 memorial lectures to be delivered before our annual general meeting. The first lecture will be delivered by Dr. Vandana Burman, and the second lecture will be delivered by Dr. Sumit Ghosh. Dr. Vandana Burman will deliver it on bioinformatics, and Dr. Ghosh will be delivering on the COVID-19 situation. Welcome, Dr. Bharati.
Are you seeing Dr. Anil Kumar Ghosh?
Right. Please present it.
Okay. Now I am. This is the inaugural speech of Dr. Anil Kumar Ghosh, the President of Institute of Science and Education. It was a WhatsApp voice message. Please listen to his inaugural lecture.
The voice is not coming. Dr.
Sorry?
Burman, voice is not coming.
Voice is not coming.
No.
Okay, just
And the photo is also not coming. Please again, please. I think it is muted. It was muted, I think. Please try again.
just one.
Thank you.
Okay, photo is coming.
It is being presented. Please wait a few minutes. Now the photo is coming.
Okay.
Photo is coming. Now audio has not yet come. Your audio is muted. Please unmute it. Your audio is muted. Presentation audio is muted. Please unmute it.
No, it is now.
It has not yet come. Let's wait. No, our system shows that it is still coming.
Culture,
Yes.
honorable invited guests.
Now it was coming. Please present.
Honorable Vice President Dr. Sapna Mukherjee, Honorable Secretary Professor Santam Das, respected advisors, distinguished speakers of today's memorial lecture, eminent members of the Executive Council, esteemed members of the Institute of Science, Education and Culture, honorable invitees and guests. It is indeed a pleasure for me to be privileged to say a few words on the occasion of the inauguration of the 40th Annual General Meeting virtual of the Institute of Science, Education and Culture on the video platform due to unprecedented COVID-19 pandemic situation. I, on behalf of myself and the entire family of ISEC, deeply mourn the unfortunate demise of some of our beloved members in the recent past and pray to Almighty that their immortal souls rest in eternal peace in heaven. I am tempted to give a brief account of the Institute of Science, Education and Culture. It was established in the year 1980 by a galaxy of scientists, professors, under the able guidance and dynamic leadership of great Dr. Muralimohan Vishwas, past life secretary of the Institute, with the primary objective to promote, propagate, and advance the knowledge of science in society as a non-profit registered professional scientific society with more than 400 life members from India, USA, UK, Germany, Australia, corporate members, institutional members, large number of subscribers covering renowned institutions, national research centers, national library, and almost all the leading universities of India. Among its various activities, ISEC publishes an international standard journal named Indian Science Cruiser, 6 issues a year. ISEC is financially supported by the Government of India and Government of West Bengal. It organizes national seminars on science, engineering, technology, medicine, industry, construction, as also environment, global warming, climate change, ecology, sustainable development, non-conventional sources of energy, education as also lies an achievement of iconic scientific personality of national and international repute. It organizes memorial lectures such as Namanita Satish Chandra Vishwas Memorial Lecture, Dr. Murali Mohan Vishwas Memorial Lecture, as we have done this time. The Namanita Satish Chandra Vishwas Memorial Lecture was instituted by the illustrious son, late Dr. Murali Mohan Vishwas, on his donation to the institute. Late Namanita Satish Chandra Vishwas were saintly persons, parents of late Dr. Murali Mohan Vishwas. They were deeply devoted to Ramakrishna Mission and were initiated by Swami Virajananda Maharaj, the last direct disciple of Sri Sri Ramakrishna Dev. Late Dr. Murali Mohan Vishwas was also an ardent follower of Ramakrishna Mission, also initiated by Swami Virajananda Maharaj, late President of Ramakrishna Mission. In fond remembrance and loving memory of the departed soul, ISEC instituted the memorial lecture Dr. Murali Mohan Vishwas after his demise to perpetuate his memory. I, on behalf of myself and the members of the family of ISEC, pay our respectful homage, glowing tribute, and steadfast ovations to the memory of the departed soul. I would like to bring to the kind notice of the members of the ISEC, and we have decided to hold the first virtual meeting of the newly elected Executive Council. Date itself after an interval of minutes after election is over, in order to elect the office bearers of the council and also of the committee. I would also like to add that the audited statement of account, which has not yet been ready in view of the present situation, will be uploaded in the computer as and when ready, after being approved by the Executive Council for the interested members to know. I would like to congratulate in advance the newly elected members of the Executive Council and other office bearers. I earnestly request the honorable members of ISEC to extend their all-out support, assistance and cooperation to the newly formed Executive Council in the discharge of its bounden duty with fullest satisfaction to the members of ISEC. With these few words, I would like to welcome you all in the virtual meeting and inaugurate the 40th Annual General Virtual Meeting on the video platform. Thank you all, wishing you all and your family very, very good, happy and peaceful festive days ahead.
I personally thank the President, Professor Dr. Anil Kumar Ghosh, for delivering this recorded lecture. And now I will request Dr. Swapna Mukherjee, Vice President of the Institute of Science, Education and Culture, to kindly address this audience. Dr. Mukherjee, please.
Thank you very much for inviting me to speak in the AGM. We are missing very much our president, who will so elaborately speak on the organization in each meeting. However, I am thankful to Bandana for arranging to communicate our president's talk. I wish everybody a good afternoon, and I am wishing all the participants to enjoy the lectures given by Vandana and Samilba. First of all, I must appreciate our very dynamic secretary, Dr. Shantanu Das, who is organizing and making alive the existence of ISEC in so many different ways. I want to appreciate it and clapping for the same. And I really feel sorry that so many members are not able to join, like Shivaji, like Chitragut Babu, and so many things. It will be a very big gathering if they could join. And I wish our ISEC will be in good hands, Shantanu Das, who will steer the organization in right direction. Thank you.
Dr. Mukherjee, have you completed?
আমার হয়ে যাচ্ছে?
Oh, you have already finished. Okay. So, thanks to Dr. Mukherjee for addressing this gathering on the occasion of this 40th annual general meeting of the Institute of Science, Education and Culture. Now, it is the time for the memorial lecture. We have the first memorial lecture in the name of 7th Nobonita Satish Chandra Bishwas Memorial Lecture 2020. And this lecture will be delivered by Dr. Vandana Bormon, a member of faculty of Electronics and Communication Engineering Department, College, Kolkata, and the Executive Council member of the ISEC. Her topic is bioinformatics. Now, I will read through her brief vita for the benefit of the persons concerned who are following this proceeding. Dr. Vandana Bormon is currently faculty in Electronics and Communication Engineering Department in Kolkata Government Engineering College. She did her Bachelor of Engineering degree in Instrumentation Engineering and Master of Engineering in Electronics and Telecommunication Engineering from Jadavpur University in the year 1997 and 2000 respectively. After passing bachelor degree, he joined as a lecturer in the Institute of Mining in Raniganj, Bardwan, West Bengal, and resigned from there for pursuing master's degree. After passing master's, he joined as a lecturer in Electronics and Communication Engineering Department in Jalpaiguri Government Engineering College. He is doing her research work regularly and co-authored several research papers in different national and international journals and conference. Dr. Bormon is also a regular reviewer of some national and international journals and conferences. He is a life fellow of the Association of Engineers India, Institute of Science, Education and Culture, Institute of Electronics and Telecommunication Engineers India. He is also a life fellow of the Institution of Engineers India and Indian Society for Technical Education. Also, he is a life member of Paschim Banga Vigyan Manch, West Bengal. He is also the life member of International Association of Engineers, Hong Kong. He is elected as the Executive Council member in ISEC as well as in the Association of Engineers India. Dr. Bormon acted as chairperson in different conferences and also he was a member of different committees. He obtained her PhD in Engineering and Technology from the University of Kolkata, West Bengal, in the field of computer science and engineering. Dr. Bormon's research interests include bioinformatics, computational biology, digital and medical image analysis and processing, big data and cloud source data analysis, machine learning, etc. Recently, he started collaborative research work with Professor Shantanu Das, Professor and Head of the Mechanical Engineering Department of Kolkata Government Engineering College, to impart the characteristics of grinding heat and surface using image analysis data. With this data, now I request Dr. Bormon to kindly present please. Dr. Bormon, please.
Thank you, sir. Am I audible? Am I audible? Am I audible?
Yes.
Okay.
Yes, you are audible. Yes.
Thank you, sir, for your nice introduction. I am very happy today that ISEC has invited me to deliver the lecture, that is the 7th Nobonita Satish Chandra Memorial Lecture 2020. I then I solely thank to the ISEC Seminar Committee, Vice President and Secretary, and all the dignitaries of the ISEC. Now I am presenting my lecture. Are you seeing my slides?
Not yet
It
come.
has opened. It has opened. The slides have not come yet.
Not yet come.
Slide is not coming. Okay. Just 1 minute. Is it coming? Are you seeing it?
It is coming. Just wait a minute.
Yes.
Now it is visible. Please present the slide.
Okay.
Make it full screen. Slide presentation from the beginning. Yes. From the beginning. Right.
Okay.
Okay. Now it is visible. Please present.
Is it clear?
Yes.
As in introduction section, Dr. Shantanu Das, the Secretary of ISEC, has told that today is the 201st birthday of Pandit Ishwar Chandra Vidyasagar, and who was the pioneer of women education. Because of his effort and struggles, all the girls in every family deserves education. And only because of this person, this great person, I am here today to deliver the lecture. I bow my head down in front of Pandit Ishwar Chandra Vidyasagar. Now I am starting my lecture. I will discuss my lecture on bioinformatics. First, I shall tell some basics on bioinformatics. After that, I shall discuss a case study to infer that is there any link between HIV-1 virus infection and cancer? A computational interference. So, first, what is bioinformatics? Bioinformatics is an interdisciplinary subject. Interdisciplinary subject means the subject associated with different subject by which one can investigate the solution of a problem. And the bioinformatics is also a management information system for molecular biology, and it does global analysis. Management information system, that means it can collect data, it stores data, it organizes the data set, and global analysis means it analyzes the data set globally. That means a huge analysis is done using bioinformatics. And we know the bioinformatics deals with biological data first, and biological system is a dynamic system. Why it is dynamic? That the biological system is changing its characteristics and it interacting with other systems or other organisms with time. So we can say the biological system is a dynamic system. And bioinformatics is a conceptualization of biological molecules, what I told just. Informatics techniques. Informatic techniques part, it deals with some technical computation related to mathematics, statistical analysis, and understand and organize information from large-scale data. Here, the big data analysis takes a major role. One should remember that we should not confuse bioinformatics with genetic algorithm. The genetic algorithm is the concept of Charles Darwin's theory, that is the adaptability of a human being, of a biological system with the change. Next, what is central dogma? The central dogma is called the backbone of molecular biology. the list of gene constitute chromosome, and chromosome is formed by deoxyribonucleic acid, that is DNA. DNA is double helix structure. It consists of two strands. One strand is overlap with on other strand. The starting point and end point of one strand is opposite to the starting point and end point of other strand. And DNA nucleobase are ATGC, these are amino acid, A for adenine, T thymine, guanine, and cytosine. A and T makes hydrogen bond, double hydrogen bond to make a bond A and T. G and C creates are attached with triple hydrogen bond. By the process replication, a new DNA form an existing DNA, and this is also an enzymatic process. After the second step of the transcendental dogma is the transcription. By the process transcription, the single helix ribonucleic acid, that is RNA, is formed. The base of the nucleobase of RNA is AUGC. You can see only U is replaced from T. Here A and U make the double hydrogen bond, and G and C makes triple hydrogen bond. RNA replication is also a process by which a new RNA is formed from a existing RNA. From a part of RNA, the messenger RNA is formed. This is a part, so its size is smaller than this RNA, previous RNA. So messenger RNA carries the information, and from a part of the messenger RNA, the protein is synthesized, and this process is called the translation. And this is a large sequence, and proteins are involved in almost all biological activities. Those are structural or maybe enzymatic. And proteins are the sequences of amino acid. There are 20 different types of amino acids that can be combined to make a protein. Now, what is microarray? What is microarray technology? DNA microarray is a multiplex technology used in molecular biology, and DNA microarray is commonly known as the gene chip, DNA chip, or biochip. And this is also a collection of microscopic DNA spot attached to a solid surface. Now I am describing the method of microarray technology. What is happened there? Suppose this is sample 1. This is the normal cell without disease, normal condition, genomic DNA, cell with genomic DNA in condition A. And here is cell with genomic DNA in test condition. This is maybe a diseased cell. By the process isolation, that is the transcription process, mRNA produced, the messenger RNA. The reverse transcription is a process which is the reverse to the process transcription. In this process, the reverse transcription, which is an enzyme, is taking part to create the cDNA, that is the complementary DNA. I shall discuss this elaborately while I will discuss my case study on HIV. And then labeling process is done. Labeling is a process in which the fluorescent cyanine dyes is poured into the sample. So here the dye is CY3, that is the fluorescent greenish yellow colored dye, and CY5 is the fluorescent red color. These two dyes are poured into the cDNA of the sample data set. After that, hybridization of these two dyes are made, and with a very highly sensitive laser, the scanning process is made to extract the image of microarray. This image has different colored spots. Each spot contains the genomic DNA that represent a unique gene. If I'm magnifying this picture here, it will show that in the row, in the column wise, these are the samples value, sample annotation, and in the row wise, it is the gene annotation, that is the name of the genes, and each box of the matrix shows the gene expression level. How this expression level comes? The cDNA microarray, suppose M is equal to the log2 value ratio of log2 value of R and G. R is the disease cell expression value, and G is the normal cell expression value. And when M is less than 0, that means the green colored value is higher than the red color, and M is greater than 0 means red color, that is red color dye values are greater than the green colored value. So these are the different parameters by we can say the expression level changes from one sample to another sample. When normal cell expression value goes high and the disease cell is low, that is changes in disease gene expression level is lower than the normal cell. Here we can say the gene is upregulated. The gene expression gene is downregulated. Gene expression value becomes lower. When it is equal, it is defined by the yellow color. So no, it means here is no changes between the expression level value of gene. And when M is red color, that means the disease cell expression gene expression value is higher than the normal cell expression value. It means upregulation happens there. When that genome data or biological data is a huge data set, so concept of data warehousing comes here. The open source data warehousing solution, that is, for example, Intermine and Biomart have been created. This is the website where from one get the data set of microarray data set. One can get the microarray data. What are the importance of microarrays in bioinformatics? Because microarray contains huge number of genes with their expression value. It can compare gene expression value, and it examines the genomics, that is the expression in one sample on genome-wide scale. And the bioinformatics, which deals with the microarray data, the microarray technology, microarray data performs a M data mining. So it does not perform a M-less data mining. It means it always investigates a solution, a perfect solution of what is happened or what will happen. There are different two types of microarray. One is spotted array, which I show you in previous slide. That is log2 of intensity R and G. Another is Affymetrix. Here what happens, the mismatching is done. Perfect match intensity and mismatch intensity. Perfect match intensity is PM and mismatch intensity means the MM. After that, the experimental data preparation is done in microarray technology. One method is cleaning and another method is normalization. In cleaning process, image processing takes a huge part. I shall discuss this later. Another is filtering, missing value estimation. The gene expression value which has very low value, which is not taken in consideration. And filtering the expression value with null value, that is with zero value, it is not also taken in consideration. It is sorted out. And the gene with missing value estimation, estimation is done for missing values, and these genes are also eliminated from the list. After this cleaning process, the experimental data set comes for investigation. Another is normalization. This normalization is a method to scale the gene expression value. Here, it removes sources of systematic variation, and maybe some background noise attached with the microdata set, or other intensity value can be attached with the data set. For this purpose, normalization is done, and with the normalization process, a scaled data is obtained with mean value scale data with mean value 0 and standard deviation with 1. And I told in last slide about the cDNA. The cDNA reverse transcription process, by the process reverse transcription, cDNA is produced. But naturally, the cDNA is also naturally be produced by the retrovirus. What type of retrovirus? That is the HIV1, HIV2, simian immunodeficiency virus, etc. And then integrated into the host's genome, where it creates a provirus. What does it do? That HIV1, this is a retrovirus. It carries the reverse transcriptase RNA enzyme. It is an enzyme, and when it attacks the host cell, it normally attacks the immuno cell of the host, and when it combines with the DNA or RNA of the host cell, it will produce the provirus. This provirus again attack another cell or tissue of the host cell, so that one disease grows. So microarray are used in research laboratory in the form of printing or spotted of probes onto surface, which is a glass microscopic slide, as this is an attractive medium of and economical. Actually, the dyes, if I see the show you the previous slide, here the solid surface hybridization, after hybridization it is poured in the solid surface. This is a fluorescent based glass material. So this is economical and also the attractive medium by which colors becomes very prominent to take the images from there. Now the concept of gene regulatory network and genetic network. Gene regulatory network, GRN, or the genetic network is a bond of DNA segments in a cell which interact with each other indirectly through their RNA protein expression products and with other substances in that cell. This is a block diagram of gene regulatory network. First, suppose two DNAs are there, and by the transcriptional process, the DNA, from DNA, mRNA are produced. This is the transcription process, and by the process of translation, proteins are produced. These proteins, sometimes some errors are attached, or some erroneous proteins are produced sometimes, and these proteins, after production of this protein synthesis, a post-translational modification happens. By this post-translational modification, a modified protein produced. And one more thing, otherwise one protein is combined, when combined with other substances of the cell, that is, it may be combined with different enzymes or other lipids, and it generates a protein complex. So it is seen that in this process there may be a huge amount of protein may be produced. So to keep the system stability, a negative feedback occurs there. It is shown by this line is the reverse. It is the negative feedback, and this is named as the transcriptional regression. Okay. This is the biological concept of GRN, and this is how the interaction made. And to make the gene regulatory network computationally, the artificial neural network takes an important role. And also, also one can construct GRN with other process computationally, or several web tools are there to make the GRN. And GRN is constructed in intercluster or intracluster. Intercluster and intracluster. Before discussion of intercluster, I want to just take one line or two lines about the cluster. The clustering process is a process. Clustering is a process by which same kind of element are grouped. Same kind of, maybe here biomolecules, same kind of biomolecules, that may be genes, maybe DNAs, maybe RNAs, are grouped and create a cluster. When one cluster interact with other cluster to make a network, so it is called the intercluster. And when the biomolecules within a cluster interact with each other, that is called the intracluster network. So the concept of one thing, the reverse engineering. The concept of reverse engineering is used in human DNA as learning tool. What is reverse engineering? Reverse engineering is a process by which one can investigate, or one can construct, or one can unconstruct the process of system. So it is the software programming, coding, and finding logic. Now another thing is protein-protein interaction, that is the PPI network. The PPI network, it is the intentional physical contact established between two or more proteins, and PPIN is the result of biochemical events and the electrostatic force. Proteins acts as enzymatic acts as enzyme, or it can be a translation or transfer process, or it can interact with other biomolecule. So now I'm showing a basic block diagram of basic network structure. Here, node V1, V2, V3, V4, V5 are the proteins, and A1, A2, A3, A4, A5, these are the edges. Edges means the interaction line. And this is a simple way, a simple diagram, that V1 and V4 is connected by this edge A3, and I have not put any direction. So this is two-way interaction, or otherwise undirectional. This is undirectional network. If I put an arrow here, it means this V1 force or make the interaction this. So in that case, the interaction will be V1 to V4. And the DNA sequence of a gene encodes amino acid of protein. Here, ribosome produce protein using mRNA as template. I told the protein function, the antibody, enzyme, messenger, structure, component, transport, storage, this kind of thing. And here bioinformatics also helps. bioinformatics helps to analyze the structure, functions, and the evolution of several proteins. After that, I talk about the KEGG pathway. The KEGG is the full form of KEGG is Kaito Encyclopedia of Genes and Genomes, or genome, and it is the knowledge base of building blocks. So this KEGG pathway is one of the metabolic pathway. And what is the metabolic pathway? Metabolic pathway is a subnetwork of a complete network. What is complete network? Suppose in human body, several enzymatic reactions are after eating or after during respiration. Several enzymatic process reactions happens in human body. So all these are the complete network. The system is attached with in a different way, linking in different way to create a complete network. And metabolic pathway is the subnetwork of a complete network. And to obtain the metabolic, we are the I am a technical person. I don't have a wet lab, that is the biological lab, in which microarray analysis is done or study of genome structure. I should take the assistance of the web tool to understand or to get the KEGG pathway analysis. I performed David web tool. This is a web tool. This is available web source to get the KEGG pathways. The KEGG metabolic pathway presents the knowledge of molecular interaction, reaction, and relation works to obtain metabolism, gene information processing, environmental information processing, cellular process, organismal system. What is organismal system? This is high-level properties of a complex organisms arise from interaction. That is the organismal system. And also metabolic pathway investigate the human disease, and it also help the drug development to develop the drug of a particular disease. From here, the concept of technology comes. First, data. I shall tell what is data and what is information. What is the data? Data is the facts and statistics collected in raw form for analysis or reference from different sources. Data comes from different sources, and information is the processed data, processed and organized useful data, which is the experimental data. The words experiment data I told when I was discussing microarray technology and the process normalization and cleaning. After cleaning and normalization, experimental data comes from the microarray data. And this is the raw data set. After processing, information comes. That is the experimental data comes. The biological study examines individual system in detail and it compares them with a related study. So it deals with a large genome data set of biomolecules and performs difference processing to obtain information of the collected data. And I told that global analysis is done by the bioinformatics, and it is performed to uncover common principles and to highlight the novel features of the system. Now, I told the analysis. Now, the concept of analysis and analytics, I should explain. The first in the before analysis was the concept of analytics was not there. The analysis only analysis was there. Now, analysis and analytics is to think in terms of past and future. That analysis means the separation of whole into its complex component parts. That is, it looks into the history So it looks backward, which and what was happened, or what was happened before. And data analysis does this task collection, organization, storage, and type of tool techniques were used. The term analytics, these term analytics which investigate, which shows the path of future. It is the method of logical analysis. So analysis is a component of this analytics, and it looks forward to model the future or to predict a result. Data analytics include my data mining, machine learning, data warehousing, crowdsourcing, and it deals with different analytics. Data warehousing, crowdsourcing, these are the part of data science. Data mining, all these are the part of data science. It is a very lucrative area of computer scientist, information technology. Now, before this slide, I was just telling the basics of this case study. Now, my case study, is there any link between HIV-1 virus infection and cancer? It is a question. And I have done a research work with my students computationally to infer a link between these two diseases. I told the concept of retrovirus, and HIV, that is a human immunodeficiency virus, is a retrovirus. It has the reverse transcriptase enzyme, and they transcribed RNA into the DNA after entering a human cell. After that, DNA also exhibit the chromosomal DNA of host cell, and this host cell DNA then becomes a provirus, and this provirus again interact with other DNA or other cell tissues, and thus this disease grows. Previously, it is found that HIV is developed from a chimpanzee virus, and that was transferred in human body in 1920 in West Africa. HIV is transmitted mainly sexual mode, either hetero or homo, body fluids, needle sharing, feto-placental transmission, that is mothers to fetus, it also transmitted through the breastfeeding, blood transfusion, and there are two types of HIV infection. First one is HIV-1, HIV-2, but HIV-1 infection is very huge rather than the HIV-2 infection. And my case study is with HIV-1 infection. And the HIV infection first attacked the immune cell of the human body, that is the cell differentiation, CD4 cell, CD4, CD2 T cell. These are the immune cell of human body. This lies beneath the skin and in the dendritic cell, and it infects first the immune cell CD4 and CD8 T cell, and because of this infection, HIV infection, the affected person's immunity gets progressively weaker and weaker, and at the last stage, the acquired immunodeficiency, that is AIDS, happens for that affected person. Now, since last 6 or 7 months, we are very aware of about the pandemic situation. But previously, HIV infection was considered an epidemic, but it is considered as pandemic since late 20th century. As reported by the WHO, that is World Health Organization, the HIV/AIDS pandemic has killed more than 35 million person since 1981. So it's a very huge number who dies because of this HIV infection. For my case study, I took the sample data set, that is GSE 6740. It is available, is a open platform, this website, and HIV microarray data. This is a gene expression omnibus database, and this data set includes untreated HIV infected individuals at different clinical stages. And what stages? That is acute, chronic, non-progressive, and uninfected stage. So this data set shows the disease progression in each stage. The data set consists of 20,283 genes and 40 samples. Among these 40 samples, 10 samples are early HIV infection, that is acute stage, and 10 samples are chronic HIV-1 infection stage, and 10 samples of HIV-1 non-progressor, that is the stage of low or undetectable viral load stage, and 10 are uninfected samples. For my study, I have not taken these samples, these uninfected samples. I have done my study with these 3 types of samples to understand or to analyze the disease progression in human body. Now, what is the proposed method of this research work? Sample data set is the raw data set, and data pre-processing is important, and data processing is done to obtain the experimental data, that is the valid experimental data, to obtain a valid experimental result. At first, the background noise are eliminated. Background noise that may become the location specific adjustment, background adjustment, light intensity value, etc. And then the perfect match, that is PM, what I told, that is the Affymetrix data, perfect match intensity depending on the mean mismatch intensity. In step this is the step 2 is to find the expression matrix. In step 3, after computing the expression value, a scaled normalization, that is the normalization is done to scale the experimental data to adjust the each array in the data set, where mean value becomes 0, matrix mean value of the data matrix becomes 0, and the standard deviation value becomes 1. And low detection level is also removed from the data set. And this function, for this, to eliminate the low detection level, the Wilkerson signed rank-based gene expression task is done, and by which perfectly matched probes are discriminated from the mismatched probe. After this pre-processing, 14,869 genes are sorted out, and these are called as the present call from the data set of 22,283 genes. Now the concept of differentially expressed gene. What is differentially expressed gene? The differentially.
Dr. Barman,
Yes.
I will request to please wind down within 5 minutes.
Okay. Are you seeing the slides? Hello?
Please make it good, please.
Oh, okay. So I'm just doing very quickly, delivering the lecture. Now, the differentially expression, as I told before, that disease, when gene expression level goes high in red or diseased by expression value, it is called the upregulated, and when decreased, it is called the downregulated. To get the DGs, the t-test, Wills t-test is used. This is the equation for Wills t-test, and Wilcox's t-test. Here the number of gene in both samples are equal, so N1 is equal to N2, but mean value will be different, and t-score value is obtained from this equation, and p-value, and this is a hypothesis test, and p-value is taken, this is the critical value. Here the critical value is taken 0.01. Lower the p-value is called the highly significant or gene expressed, differentially expressed gene. So first I made the pair of three pairs: acute, chronic, chronic non-progressive, and progressive acute. From these three pairs, I got this number of DGs, and these DGs are further investigated to get the co-expression network. For co-expression network, to get the co-expression network, adjacency matrix is calculated by finding the correlation between the genes, and from the adjacency matrix, the weighted value of co-expression, differentially co-expression genes comes out, and after that, the co-expression network. Just I'm going to the next slide. Here is the some equations by which the weight function are obtained from that differentially expressed gene, and this is a topological overlap map, that is the Tom similarity matrix. The similarity between the co-expression value, the Tom analysis is done, and similarity and dissimilarity. After performing the dissimilarity and similarity, it is that the 151 genes are found for acute chronic, and 133 for chronic non-progressor, and 220 genes comes out for the non-progressor and acute differential co-expression network. And this is the co-expression network. This is for the acute chronic, and this is for the chronic non-progressor, and this co-expression network for the non-progression and acute. After that, dynamic tree cutting method is done. Dynamic tree cutting is nothing but a presentation of hierarchical clustering. The hierarchical clustering is a process by which distance between the samples are taken, and which when the distance becomes high, so it may be two different clusters. So here the dynamic tree cutting. This is this is the structure by which first one minimum point is taken consideration, by which these are the clusters or different modules of cluster modules of this weighted differentially co-expressed gene network. And resultant analysis. This is the resultant analysis. I told actually I'm sorry that I couldn't tell the Tom analysis very describedly. So these are the diagram. These lines means the similarity, and these are the dissimilarity. After that, the acute chronic eigen network. You can see the diagonal values are 1 for eigen network, and this are the diagonal part of chronic non-progressor and non-acute eigen network. And after before this up to this slide, this was the analysis, and then we are all aware about the cancer disease. So the cancer disease happens when human body grows uncontrollably. So uncontrollably, that is rapid gene mutation occurred when affected. It may be external, but disease side effect of some disease, or some tumor and rapidly growth of tumor cell, and the HIV disease destroys the immunity system of an affected person. So the cancer may occur for HIV affected person. It may occur because immunity, the less immune person. And after this, the cake pathway analysis is done for the genes what I obtained by my analysis. Here it is seen here I take the assistance of web tool, David tool, web tool David tool, and found that 36 genes which are listed in HIV-1 infection are significant also cancer disease. You can say this gene is significant in transcription misregulation in cancer. Hepatomic cell, I just put the red color which for the cancer significant term. This gene is in HIV infection as well as pathway gene cancer, viral carcinogenesis, pancreatic failure, there is bladder cancer, leukemia, cancer, this is the cell lung cancer, and this gene, so 36 genes is somehow linked with the cancer. This is also pathways in cancer, this gene. So 36 genes are listed here. So more or less these are the significant in cancer disease also. So in conclusion, I can say that in my presentation I have done a comprehensive analysis of geo data set of HIV infected and then cake pathway and significant genes I found co-expressed. I constructed the weighted differentially co-expressed gene network, then cake pathway analysis, and lastly found the 36 genes are significant in also in different cancer disease. Furthermore, it can actually I have found the 36. Furthermore, in future, one can find the drug targets. That means which genes can be useful for finding the drug of cancer disease also from these 36 genes. And lastly, I can say that the gene is a program. Actually, in bioinformatics, gene is a program which create codes to run the central dogma. Central dogma, that is a system to run the central dogma, and this is main thing that we are doing coding by our own language. Different softwares are used to investigate this. MATLAB is very useful software. One statistical software is R. Python is now taken for this, and other tools to understand the protein. Cytoscape is a software. StringDB is a web tool to get the protein interaction network. And these are the references. Only this paper I have taken to get the differentially weighted co-expression network, differentially co-expression network. This paper, it was a very relevant paper I have taken them. But other papers of mine paper, here mathematical modeling of gene expression as a dynamic system, it was a conference paper. This is the paper by which was based on finding the genetic network. This is the minimization of data matrix. These are the whether data macro data, time series data is linear or non-linear. These are the after that computational model of metallic pathway. So these are the paper, and the today's lecture on which I made. It is this work is also communicated to a book chapter as a book chapter in Elsevier. It is with the publisher now. It will be published very soon. Preprint is ready. So lastly, I thank you all, and thank you all for your patience. My best regards to the seminar committee, to all members of Executive Council of ISEC. Thank you all. Thanks you for your patience. Now over to
Thank you, Dr. Barman, for a nice presentation. Really, it is a new area, too much technical also. And now, as we are running a little late, I can invite few questions only, one or two questions from the audience, if there is any. If there is any question, please ask. It is the session for question and answer. Any person from the audience? Any question? Okay, if there is no question, I am just putting one question as a layman in this area. Can this gene mapping, etc., will be applied for COVID-19 situation also to find out the vaccine, etc.?
Yes. Competition, actually, we are doing the dry lab investigation. That is the software-based application. And wet lab verification, that is the biological application, is very important to find out a vaccine. Okay. So, as a bioinformatics, as a person in the field of bioinformatics research field, I can say we can help the biologists to find out some significant genes which are related to or significant to the COVID-19 disease. By these genes, one can find out the drug target and drug also, that is a vaccine also.
Thank you. Any other questions, please? If there is no, then I close this first memorial lecture, 7th Nabolika Satiswara Vishwas Memorial Lecture 2020. And I especially thank again Dr. Vandana Barman for nicely outlining that area of our work in this particular typically bioinformatics area. Now I come to the next lecture, memorial lecture, that is 4th Dr. Murali Mohan Vishwas Memorial Lecture 2020, and that will be delivered by Professor Dr. Sumit Ghosh. And now I do have the privilege to read out the bright and large number of activities in which Professor Dr. Sumit Ghosh is actively involved in. Professor Dr. Sumit Ghosh is an MSc and PhD, and he is a fellow of Alexander von Humboldt Institute, and also a fellow of Max Planck Gesellschaft, Germany, and he is a consultant medical physician. He has done doctorate degree from Calcutta University in physics in 1965. I request others to please make the audio a little muted. Thank you. So, Dr. Ghosh has completed doctorate degree from Calcutta University in physics in 1965. He was a research fellow of Government of West Bengal and Government of India during 1958 to 1962. He was a postdoctoral research fellow of Alexander von Humboldt Institute and Max Planck Gesellschaft at Max Planck Institute for Biophysics at Frankfurt or Main, Germany. During this research work, special stress was given on biophysics, dosimetry, and radiation treatment with high energy gamma rays and electron beams at the Peterson Department of Max Planck Institute for Biophysics. He obtained a certificate which entitled One War and Specialist in Radiation in Germany. He attended 4th semester in Wuhan, Ingang, Goethe University, Frankfurt, laying on biophysics, nuclear physics, and physiology. He attended the courses on safety aspects in medical uses of radiation conducted by DRC, Department of Atomic Energy, Government of India, in 1967. He He was a lecturer in physics at Joypuria College, Kolkata during 1967-68. Then he worked as a school officer, Government of India, at the Physics Department of Presidency College, Calcutta during 1967. He is a physical medical college. He is a physicist at Medical College Hospital, Calcutta during 1967 to 1992. He is also a medical physicist at Chittorganj National Cancer Institute, Calcutta during 1992 to 1994. He was also the head of the Department of Medical Physics, Cancer Center and Welfare Home, Thakurpur, Calcutta. Also, he is a medical physicist of Regional Cancer Center, Coachbihar, and he was there during 1994 to 96. He was also a technical director of radiotherapy, Maharashtra Cancer Research and Welfare Center, Maharashtra, West Bengal, during 1998 to 2001. He was also a medical physicist at Regional Cancer Center, Coachbihar, at present since August 2001. He is recognized as Radiation Safety Officer by Atomic Energy Regulatory Board. Last renewal was done in 2011. He attended DRAS workshop on quality assurance and surveillance in diagnostic radiology at AMIO, Bangalore in 1989. He was also the convener of Radiation Hazard Committee of the Government of West Bengal. He is also a former honorary visiting consultant in nuclear medicine of BR Singh Hospital, Eastern Railway, Government of India. Formerly, he was honorary visiting consultant in medical physics of SCS Marwari Hospital, Calcutta. He represented National Institute for Biophysics at International Radiology Congress at Rome, Italy. So it is really a very good privilege on my side to read this biodata, but this biodata is not yet completed. I continue. He is also one extra-mural lecturer at the University College of Medicine, Calcutta University, during 1968 to 1990, and he is teaching there on CMRT, DMRT, and MD radiotherapy and MD radiology diagnostic courses. Presently, he is the professor of radio diagnostic technology courses under State Medical Faculty, West Bengal, and DMD radiology courses at Vivekananda Institute of Medical Sciences, Ramakrishna Mission, Ramakrishna, Calcutta. He is also a professor of Institute of Radiology and Medical Imaging in Radiology Diagnostic Technical courses. Formerly, he was the examiner of Calcutta University for DORD or DMRT and GST examination. Presently, he is the examiner of State Medical Faculty, West Bengal. He was formerly the examiner of Bombay University. He was also referee of research projects of ICMR. He published more than 20 papers in national and international journals. He obtained a Lifetime Achievement Award for the development of medical physics from Association of Medical Physicists of India in 2006. Finally, he was felicitated by Department of Radiotherapy, MCH Kolkata, and Association of Radiation Oncologists of India for his continued services in medical education and commitment to cancer care 2010. So now I request Professor Dr. Ghosh to please present his quote, Dr. Murali Mohan Vishwas Memorial Lecture 2006. Professor Ghosh, please.
Thank you, Mr. Chairman, and the respected members of this audience. You see, for this long introduction, it was not required. I am a simple man. I am just working for the last 60 years in this line and have spent my life for the benefit of the people. Now, since I am at the age of 86, I am not ready to take all these online lectures and etc. It is only for Professor Murali Mohan Vishwas. I hope I could reminiscence a bit something about him. That's why I accepted it. Professor Murali Mohan Vishwas. You know him mainly as Professor of CTTI, but I am going back to an olden days. I know him from 1995. Sorry, it is in 1955. It was in 1955 when I joined Presidency College as a research scholar. When Professor Murali Mohan Vishwas was a research scholar there, and he joined there about 3 years back. We were 4 persons there. That's a group of 4 persons. One is Dr. Kamal Rai. He is no more there. He is dead. Another is Professor Murali Mohan. Second man was Professor Murali Mohan Vishwas. Third man was Professor Olup Acharya. I have lost track of him. I do not know where he is now. And I was the fourth one and the youngest. I want to digress a bit here. Professor Vishwas, such a man I have never seen in my life. You know him as a CTTI professor, but I know him from his research days when he was living in a single room at the the Skole building, as a single room, and I say the very hard light. This light you have not seen. I have seen it. But in spite of that, one thing is sure, that he is the best man I have ever seen. That is, he was the friend, philosopher, and guide to me throughout my life. Even when I was in Max Planck Institute for Biophysics, he was at Max Planck Institute for Heidelberg. We never lost our contact. Till the end of his life, we were there. And such an honest man, he must be followed. He must be followed by everyone. He was the typical example of simple living and high thinking. That was the ideal. He never compromised with anything which he did not think properly. That was Professor Murali Mohan Vishwas. Now, it was he who established this at the verge, when we were at the verge of our retirement age. We thought where to go, where can we find this academic discussion. That's why we formed this society. We formed this ISEC only for that purpose, and that was our so-called Bidker Baranosa. That was the time, and it was his idea to chalk out this ISEC journal. This journal, it was named by him. It was the name that the Indian science user, that is always searching for something new. And that's why today, for the discussion, for the lecture, I have suggested which is the greatest problem of today, that is the coronavirus. I am now concentrating on that subject. I am not a clinician. I am not a virologist. I cannot tell you about the medical aspects of this one. What I can tell you is only about the social aspect of this coronavirus. Now, everybody is suggesting that there should be a social distancing between people and people. Now, tell me please one thing. Particularly, you see, I am not going to these written lectures. I am not used to this type of lectures. I always like to speak extempo, and that's why I am going out of this line. I tell you, money with the population of India, 130 crores, and I compare it with the population of America, 36 crores. Even with that population, they could not maintain the social distancing properly. The density of population in Calcutta is 24,000 people per square kilometer. And whereas in America, it is 2,700 per square mile. But in spite of that, they could not measure social distancing. And they are now advising us to maintain social distancing of 6 feet. Now tell me, 24,000 people in one place, let there be a mathematician to calculate out where can you find this space. Even the skyscrapers cannot accommodate all these persons of the 6 feet distance. That is what is happening. Now, everything, the world changes. But one thing is sure, that the people will survive. One thing is sure, people will survive. One day, you see, if you go to the history, the world has seen thousands and thousands of these epidemics and pandemics over billions of years. Devastation over devastation. And till then, the man survived. Till then, the man survived. Once you know that South Pole turned into North Pole and North Pole turned into South Pole. But even after that, the human population was there. From the period of the Neanderthals, still today, the human population is there. So there is nothing to lose heart. We will have to fight. I do not know of the 700 billion persons in this world today, how many will survive this corona attack. Last Spanish flu took a toll of 1% of the population. But I do not know what percentage of the population the present coronavirus will take. It may take 50%. Have you read the diagnosis of Professor Dr. Fauci of America? He has said, even with the best vaccination, if found, only 50% of the disease will be suppressed only suppressed, not cured. So I do not know what person should say. Even if out of the 700 billion, 400 billion dies out, but still 300 billion will be there. And they will be there and to survive, and a new society will come up. New society will come up. That society may be different from ours. That society will be completely separate, but they will be there. Social distancing, what they are mentioning nowadays. What is social distancing? That is nothing but, in other words, is social annihilation. It is nothing but social annihilation. There cannot be any social distance. Even you find the political parties are holding their mass movements. And where is their social distancing? Nobody cares. Since they have the mic, they have the TV in their control, they are suggesting everything. But we do not know in our personal life we can do it. But in spite of that, I said the world show will go on. It will never end. Only that day it will end when the sun will lose its temperature, when the sun will cool down, when there will be no more energy. Only that day the world will end. Before that, world cannot end, can never end, because then that timeline is an astronomical timeline. So we need not worry about that. We must face everything. It is like war. People cannot give up their job. People cannot give up their job. They will have to live. They will have to carve out living. They will have to feed their children. They will have to feed their family. And they must go. It is like just going to war, taking all the risk. We are going to war. We will have to fight it out. We cannot take it lying down. And that has happened for centuries. That has happened. And that will happen again. And I expect that everybody in our world will now look for the forward. So I pray to God, don't lose heart. Don't lose heart. Go forward. See, perhaps we will not be there new people will come up, the society will be formed, and they will have their own life. But world cannot end. The world so will go on. This is my appeal to you. Now, rest of the thing, I ask the chairman to continue with the rest of the lecture if there is anything. Thank you, sir.
Dr. Ghosh, have you completed? Have you completed the presentation? Hello, sir. Have you completed the presentation?
See?
হয়ে গেছে কিনা জিজ্ঞাসা কর।
কি বলছি?
Have you completed? শেষ হয়ে গেছে? Shall we continue for more time?
হ্যাঁ।
আপনি বলবেন আর
The rest of the thing, I mean, the chairman can continue with the discussion. What I have said, I have said.
Right. Thank you, sir, for the presentation and nice talk and remembering the role of Professor Murali Mohan Mishra. Now, the session is open for discussion from any person from the audience. If some question is asked, now it is open for discussion. Any member, can you please raise any question? Any person from the audience? Okay, sir, I can ask one question. That we have seen Professor Mishra staying in a small room in the ITEC Bhavan at present, and we have met you also during the half-bus opening of yours, where you have participated and opened the half-bus of Professor Mishra. So can you please throw more light on the simple life what was spent and laid by Professor Mishra? Some more light if you throw, then it will be inspiring for all of us. So can you please throw more light on the lifestyle of Professor Mishra? Sir, can you please discuss more on the simple life living by Professor Ghosh, Professor Mishra, Murali Mohan Mishra? Can you please throw more light on that? Just please unmute yourself and please tell. Please unmute and tell, sir. Okay, can you hear me? Hello? Hello?
I think he is not listening to your.
Right. Sir, can you listen me? Hello?
Hello.
Hello?
Sir, can you tell something more on the life of Professor Mishra? Can you tell more? আপনি কি প্রফেসর বিশ্বাসের লাইফের উপরে একটুখানি বেশি কিছু বলবেন, তার যে সিম্পল লাইফ স্টাইল এবং অন্যান্য যে আইডিয়াগুলো ছিল সেটার উপরে?
শোনা যাচ্ছে।
Can you please tell more on the lifestyle of Professor Mishra? Hello?
Hello?
Can you speak more on the lifestyle of Professor Mishra?
Huh?
Professor Mishra, his simple lifestyle, can you throw some more light? Can you throw more light on the lifestyle of Professor Mishra?
Mane, I have the written lectures here, but I will send you the copy. Okay?
Okay, sir.
But I did not speak from the written lecture because I am not used in speaking from the written lecture.
Okay.
This written lecture is there. I will send you the copy of that, of Professor Bishop, and of this lecture also. Okay? It is
Thank you, sir.
I have left out, I would say the 50% I have left out because I found that time was also short. I left out 50% of the things, but still the main point I do focus. The main point I have focused. That's why.
Thank you, sir, for a very nice and inspirational talk. and also you have highlighted the lifestyle and the ideology of Professor Murali Mohan Mishra. And I again put my sincere gratitude to Professor Dr. Sunil Ghosh for accepting our request to deliver the fourth Dr. Murali Mohan Mishra Memorial Lecture during the 40th Annual General Meeting of the Institute of Science, Education and Culture. Thank you again, sir, and due regards to Professor Ghosh. Now I close this memorial lecture session, and we will be having some 10 minutes break here, and just at the right time of 3:45 p.m. we will again start the annual general meeting, the 40th annual general meeting of the IPEC to transact the business session during this 40th annual general meeting. So the memorial lectures are now closed. The members, please be there in this Google Meet, and our YouTube channel that will be closed now, because only through this Google Meet connection we will continue this 40th annual general meeting to transact its regular business. Thank you all, and please have patience for another 8 minutes. So right at 3:45 p.m. we will start the 40th annual general meeting 2020. Thank you all.